Ever pondered on the science behind growing a plant in a laboratory setting, away from its natural habitat? Well, the answer lies in an intriguing biological technique known as plant tissue culture. This is a method that has revolutionized the world of biology and horticulture, enabling the growth of plants in a controlled environment. Plant tissue culture, in its simplest form, involves the use of plant cells, tissues, or organs grown in sterile conditions on a nutrient medium. It's a fascinating branch of plant science that allows the cultivation and multiplication of plants, irrespective of the season or climatic conditions. The apparatus for plant tissue culture is quite specific. The key components include a laminar airflow chamber, an autoclave, a pH meter, and a growth chamber. The laminar airflow chamber provides a sterile environment for the culture, while the autoclave is used for sterilizing the media and instruments. The pH meter helps maintain the optimum pH level in the medium, and the growth chamber provides the ideal conditions for plant growth. Now onto the crux of the matter, the process of plant tissue culture. It commences with the selection of plant material, also known as the explant. This could be a leaf, stem, or even a single cell. The chosen explant is then sterilized to eliminate any contaminants that could hinder the growth. Next, the sterilized explant is introduced into a growth medium. This medium is typically a gel-like substance enriched with nutrients, hormones, and vitamins required by the plant. Here, the explant starts to proliferate, producing a mass of undifferentiated cells known as a callus. Following this, the callus is transferred to a different medium that encourages differentiation. This medium contains specific hormones that stimulate the callus to develop into shoots and roots, forming complete plantlets. Finally, these plantlets are transferred to soil or a soil-like substrate under controlled conditions. They are gradually acclimatized to external conditions, and eventually they mature into full-grown plants that are genetically identical to the parent plant. This incredible method has numerous applications, from crop improvement and conservation of endangered species to the production of secondary metabolites and genetic engineering. It's a testament to the wonders of science and its capacity to recreate and manipulate the natural world. So the next time you see a beautiful flower blooming outside of its season, remember, it might just be the work of plant tissue culture.